Okay, so my name is Tiana, and I'm part of Group 5. We will be doing cell adhesion. Okay, so cell adhesion is the binding of cells to a surface or substrate to form tissues and organs, and they are mediated through transmembrane proteins. And what happens is extracellular portion interacts with the extracellular portions of proteins on the surface of neighboring cells. These are a list of cell adhesion molecules. We have immunoglobulins, superfamily proteins, caterins, selectins, and integrins. Cell adhesions can have two different interactions. One interaction can be homophilic, which is when cells interact with identical molecules of the surface of cells they adhere to or attach to. And heterophilic interaction is when cells interact with different molecules on the surface of the cell to which they attach to. Okay, this is a figure of different cell adhesion molecules. A shows a immunoglobulin superfamily protein with a homophilic interaction. B shows a caterin with homophilic interaction. C shows a selectin attached to a glycoprotein with a heterophilic interaction. And D shows the immunoglobulin superfamily protein attached to an integrin in a heterophilic interaction. Okay, caterins are adhesive glycoproteins found on plasma membranes. They require calcium to function, and the calcium binds to and stabilizes the conformation of caterins that allows them to mediate cell adhesion. One of the most important parts of caterins is the E caterin. E stands for epithelial, and which consists of 5 repeat domains. Okay. Selectins are glycoproteins that mediate interactions of leukocytes with endothelial cells lining blood vessels or platelets. When leukocytes invade blood vessels, they make more stable adhesions. In the figure, it shows the rolling of leukocytes, and the leukocyte has two carbohydrates which connect to the selectins, which give a signal to tell the leukocytes to keep rolling, and then the integrin attaches to the leukocyte, which tells it to stop rolling, and it firms an attachment. It firms the attachment is a firm attachment and allows invasion. Integrins are cell surface receptors that binds extracellular matrix components. They're primarily cell to ECM, which is extracellular matrix binding. They have a role in integrating the cytoskeleton with extracellular matrix. And they are important because they are primary means by which the cell The structure involves two transmembrane polypeptides, the alpha and beta subunits. The extracellular portions of the subunits interact to form binding sites for the particular extracellular matrix, depending on the subunit. The binding sites for the specific molecules of the cytoskeleton, mechanically linking the cytoskeleton and the extracellular matrix across the plasma membrane. Shown here is a fibronectin receptor, which has a binding site for a talon on the cytoplasmic side of the membrane. In this, integrins of the alpha subunit is split into two segments and held together by disulfide bonds.
So, my name is Bria, and I'll be talking about cell junctions. And cell junctions is an intercellular bridge that exists in all human cells. It consists of multi-protein complexes and provides contact between neighboring cells and the extracellular matrix, allowing them to adhere to one another and communicate with one another. There are three different types of cell junctions. One is adhesive, which is cell to cell, and another is gap junctions, which involves the chemical and electrical communication between cells, and they communicate through diffusion, but without contact of each other's extracellular fluid. Another is a tight junction, which creates impermeable seal between cells. Tight junctions act as barriers that regulate the movement of water and solutes between epithelial layers. This is a table of the three different types of junctions. We have 4 different types of adhesive junctions, which are focal, hemidesmosome, adherent junctions, and desmosome. And just explain again, the tight junction is used for sealing spaces between cells, and the gap junction is used for exchange of ions and molecules between cells. This is just an enlarged figure of cells, and we have the desmosome down there, and the hemidesmosome, and a tight junction, and the gap junction seen there. Yep. Then the apical region and the basolateral region. An adherent junction is cadherin mediated adhesive junctions that interact with actin, and it's the point of attachment between the cell surface and the cytoskeleton, and they use the beta-catenin and alpha-catenin proteins are what's used to interact with the actin, and we'll see in the figure below. So we have the A-catenin protein used to interact with the actin, and the cadherins in the middle are what are used to hold them together. This is your adherent junction. Desmosomes are points of strong adhesion between adjacent cells and tissue. and they provide structural integrity and enable cells to function as a unit to resist stress. And this is a figure of a desmosome. You got the desmogliin cadherin and desmocolin cadherin that join the two hemidesmosomes together, and on the sides you have the intermediate filament. This is a figure showing a gap junction. You have your external membrane surface, the two surfaces of the two cells, and you see that the hydrophobic channels, they are very, very close together, creating that that tight gap junction, allowing for things to pass through there. Yeah. And this is a figure of the tight junction. These are your adjoining epithelial cells, and your tight junctions are here. And we'll bring it to a closer look over here, where you have your intracellular space, and you have these transmembrane molecules that they are used to block diffusion of the transmembrane molecules. So they create these small circles, create the tight junction mechanism.
My name is Janetta, and now we're going to get into the relevance of cancer, the relevance of cell adhesion to cancer. So cancer is basically a collection of related diseases, but what all the diseases have in common is that the cells, they grow and they divide uncontrollably, and then some even begin to spread throughout the body. Cancers form solid structures as tumors, and tumors are just mass growth of tissues. There are two types of tumors, benign and malignant. A benign tumor is a localized tumor, so it grows at one site, and a malignant tumor is a tumor that grows uncontrollably, or grows and divides uncontrollably and ends up spreading through other sites or to other sites. So here this table just shows some differences between a benign and malignant tumor. So as it says, a benign tumor is small, it has a very regular shape. The tissue organization is pretty normal, and as you can see, the tumor boundary is well defined. It's a nice round shape. In contrast, the malignant tumor is large, is disorganized, and poorly defined. So you can kind of see it doesn't have a nice round shape as a benign tumor, and you can see how it would grow uncontrollably and then end up pieces breaking off and spreading. So there are 3 stages to cancer development, and the first one is initiation, the second promotion, and the third tumor progression. So right here we would have what a normal cell would look like, and then you would see one cell would mutate, and that mutated cell will go through cell proliferation, and cell proliferation is just the growth of the cell through multiplication. So you see that one turn into a whole row of the same mutated cell. And then through the tumor progression, one cell mutates over and over, and that's how the tumor progresses. And it's hard to control. So mutations cause damage to cell adhesions. So basically, they will no longer be able to function properly. They will not be able to adhere to one another. So the mutation, basically, you could say that it decreases adhesion. So the protein signals regulate, in a normal cell, a protein signal would regulate cell growth and division, and once it needed to stop, it would tell the cell to stop, but a mutated cell would continue to grow, it would ignore the signals. There are 2 groups of genes, proto-oncogenes, and these genes
are a normal gene that has the ability to become a mutated cell, and those mutated cells can transform normal cells into cancerous cells. And so the tumor suppressor genes, they slow down cell division, so they do things such as repair DNA mistakes, and also tell cells to die, which is a process called apoptosis. So basically, when the tumor suppressor genes are no longer working properly, they can cause cells to grow out of control. And so that would be like a malignant tumor. So an invasive cancer is basically a malignant tumor. It grows out of control. It has an uncontrolled growth of cells. They spread from one layer to another and ends up going through to other tissues, healthy tissues. They change cell surface proteins, and this is what I mentioned earlier. With cell adhesion, it just lessens the adhesion, so they will not be able to bind correctly. So angiogenesis is a process of growth of blood vessels. It's a normal process that happens throughout the body. And with tumor cells, basically, in order to grow, they would need the nutrients that a blood vessel would supply, and that would help it grow. So this is just an example of that. Here we have a thyroid gland, and they have injected this with cancerous cells. The cells ended up stopped growing because it didn't have the nutrients that a blood vessel would supply.
Hi, my name is Anthony, and I will be talking to you furthermore about the relevance of cell adhesion and cancer. Here we have the epithelial mesenchymal transition, otherwise known as the EMT, and it is basically when endothelial cells undergo phenotypical changes to mesenchymal cells. Cancer cells can undergo this change in order to be able to migrate throughout the body. So a cancer cell, once it has grown to the point where there is no longer cell adhesion, will break off and will undergo phenotypical changes to become a mesenchymal cell so that it may degrade the basement membrane and go into the mesoderm and then furthermore into the blood vessel to metastasize. Here is a figure showing you the normal growth of skin cells, and you see here that they start at the basal layer, and as they go up to the outer skin surface, they will lose their nuclei and begin a process called desquamation, which is basically just the peeling of skin cells. Here you see in the tumor growth that there is a uncontrolled growth of these skin cells, and they will actually break through the monolayer. And usually regular skin cells, when they get to this point, will lose their nuclei and undergo desquamation and shed the skin, and skin cells will shed. But in this process, you see that these cells are continuing to grow, and as they grow, they're losing cell adhesion and will eventually break off and metastasize. Cancer cell proliferation is basically the proliferation just basically means the steady growth of something, the continuous growth. Anchorage dependent growth, independent growth basically means that cancer cells are able to either grow on a solid surface, or they can just grow in a liquid. Density dependent growth is basically meaning that if we go back to this figure, you would see that how these cells are growing past the monolayer. They are continuing to grow even though they're already past the monolayer. Metastasis. Metastasis is when cancer cells travel to distant sites where they form tumors. Metastasis are not physically connected to their primary tumor. So once the tumor once a cancerous cell has grown to the point where cell adhesion is lost, it will break away, undergo phenotypical changes to become mesenchymal cells, and then invade the blood vessel. This process is known as metastasis, and it's 3 stages. Here you have invasion, in which they invade the blood vessel, and then you have transportation, in which they will undergo a process called intravasation, and that is the actual invasion of a blood vessel. They then will have to survive throughout the blood vessel. Some will adhere to the lining of the blood vessel, and some will undergo a process called extravasation, in which they will leave the blood vessel and then re-invade and grow. As I was saying before, intravasation, which is the process of how cancerous cells enter the blood vessel, they can enter the circulation by transmigrating either paracellularly or transcellularly. Paracellularly is basically when the cell rather than going through the actual cell itself, it will go through the junction of the cell. Transcellularly is the opposite, in which they will actually go through a cell itself in order to get to where it needs to get to. Extravasation is a process by which the neutrophil undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. And that's the process of extravasation in a regular process. You have when neutrophils respond to tissue damage, this is the process that they undergo in order to get through the endothelial wall. Cancer cells will take advantage of this process in order to enter through the endothelium. And here we have a figure explaining intravasation as well as extravasation. Here in this figure, you see that this is the site of the primary tumor, and this is the extracellular matrix, and this is the basal lamina. Once the cells have grown to the point where they lost cellular adhesion and have undergone phenotypical changes, they will degrade this, go into the extracellular matrix and degrade the extracellular matrix, go to the blood vessel and undergo a process which is known as intravasation. Cancer cells invade surrounding tissues and vessels, move through the blood vessel. Cancer cells are transported by the circulatory system to distant sites. Once they've reached a certain site, they will undergo a process called extravasation, and then the cancer cell will reinvade and grow at a new location. There was a study on siRNA, and it observed the effects of small interfering RNA, which is siRNA, targeting bone morphogenic protein receptor 2, BMPR2, on the biological activities of human liver cells and explored its mechanism. BMPR2 is critical to the growth and proliferation and invasion of cancer cells. And so what they did was they used siRNA to target this receptor, and they found that it inhibited HEPG2 proliferation and invasion and actually promoted apoptosis and blocked HEPG2 in the S phase. A separate study showing dominant negative used M6P IGF2R, which is a tumor suppressor gene that is mutated in several cancers. And they tested the potential dominant negative effects of 2 cancer-associated mutations that truncate or cut or shorten M6P IGF2R. And the results was that a transient expression of wild-type M6P IGF2R with truncated receptors led to destabilization of wild-type receptor expression. And this is cell adhesion and relevance to cancer, and I hope that it was helpful.